Okay, so here we have in text what I just went over, and also what is in the video animation of influenza virus replication. I think the important thing to remember is that that influenza has segments, and when the segments reassort into different viruses, if you have two viruses infect the same cell, two different viruses with different segments infect the same cell, there can be antigenic reassortment or genetic reassortment resulting in new viruses, okay, new pandemic style viruses. So negative strand has to go through, the other thing you have to remember is if it's negative strand, it has to go through a positive strand intermediate, and then back to negative again. Okay, so the capsid assembly occurs in the cytoplasm. Envelope proteins are synthesized in the ER, and all the envelope proteins and the capsid proteins come together at the cell membrane where the negative strand RNA segments are packaged inside the matrix, and the mature virions then bud out of the cell membrane. And we'll actually be talking about, well, here's some more influenza virus replication, but we'll actually be talking about some of the influenza antivirals that are available, like Tamiflu and some other, an older drug as well. So clicker question here, which of the following viral genomes can be translated directly into proteins? Similar to a previous question that I've asked. So go ahead and stop this, test yourself. And didn't put the answer in here, but of course the answer is positive sense RNA. Okay, so now let's talk about HIV, or retroviruses perhaps in general, but let's go ahead and use HIV because we know more about HIV even than influenza or polio or HCV. We really know quite a bit about HIV, even though it's a complex virus. So the reason retroviruses are called retroviruses is because they reverse transcribe their genome, which is single-stranded RNA. Doesn't matter if it's negative or positive sense. I think previously I might have said negative sense. Doesn't matter if it's negative or positive. I think it is positive. Into double-stranded DNA. So reverse transcriptase takes single-stranded RNA and makes double-stranded DNA out of it. It's really a complex process by which reverse transcriptase does that. And we have a slide on it, but I don't need you to know all the complexities. I just need you to know that RT, reverse transcriptase, makes double-stranded DNA from a single-stranded RNA template. And then that is integrated into the host chromosome by a protein, guess what, called integrase. Okay, so that's kind of easy to remember. The double-stranded DNA from HIV, from a retrovirus, is integrated by an enzyme called integrase. Okay? So there's simple retroviruses. These they cause tumors and leukemia. These are some of the sarcoma type of ones that we're not going to talk about, that I talk about in my graduate course. And then there are lentiviruses. HIV is a lentiviruses. These have pretty complex genomes. Not that these don't. And these cause infections that progress slowly over many years, although there are fast and slow lentiviruses too, relatively. Okay, so most of you may know, if you've read anything about history of HIV, that HIV evolved from African green monkeys. It was probably caused by practices of eating raw bushmeat, and maybe some people had cuts on their hands, and a monkey was infected with a particular virus, and it evolved to the virus itself evolved into what we know today as HIV, which causes AIDS, acquired immunodeficiency syndrome, transmitted through blood. In the early 80s, we did not have a blood test, and people were IV drug abusers and homosexuals were prevented from donating blood, and we still actually prevent IV drug users from giving blood. But we at least do have a test for HIV and all the viruses that we know about. Of course, there's lots out there that we don't know about. There's no vaccine for HIV yet, although there are many in clinical trials. There's no cure, but there are drugs, and we'll talk about antiviral drugs that work with HIV when we talk about our antivirals. So the structure of HIV, it's an enveloped virus. It actually has 2 copies of RNA in it. This looks like this says positive sense single-stranded RNA. So it is single-stranded. It doesn't matter if you know if it's positive or negative sense. The thing that you really want to know is that it carries a reverse transcriptase in the virus with it. So this is its capsid here, and then it also has a matrix around it, so it's a little bit more complicated. It also has a nucleocapsid here to protect the viral genome, single-stranded viral genome. And then it has these glycoprotein spikes here. We call these SU and TM. And in HIV, SU is called GP120, and the TM is called GP41. And these are the viral receptor for CD4 and CCR5 on T helper cells and some macrophages. So the organization of the genomes is broken up into GAG, POL, and M. So GAG encodes the nucleocapsid and the capsid and matrix proteins. Okay, so those are structural proteins, along with envelope that also encodes the viral receptor. So all these things are structural because they make proteins that provide the structure of the virus. And then POL is a group of genes that encodes the reverse transcriptase, the integrase. Remember, the integrase helps the double-stranded viral, proviral DNA integrate into the genome, the host cell genome, and then a protease. And it turns out that the protease is pretty important because we have protease inhibitors also for HIV that inhibit a protease that work actually very well for suppressing viral replication. So LTRs, don't worry too much about LTRs, but what LTRs are is they promote viral replication, and they sit on each end of the genome. I don't think I'm going to ask you any questions about LTRs. If I do, it'll be a bonus question. So if you think about LTRs, if you ever hear LTRs, think about retroviruses. So how does HIV get into cells? HIV, the SU part of HIV, or GP120, binds to the CD4 receptor of T lymphocytes. So HIV infects the quarterbacks of the immune system, remember. That's CD4 T helper cells and causes their dysfunction. It actually needs what's called a chemokine receptor, CCR5. Don't worry about that. Just know that HIV infects CD4 cells. And CD4 cells, as you already know, are very important because they secrete cytokines that tell all the other cells what to do, all the other lymphocytes what to do. So this is so close that when this happens, the envelope of the virus and the envelope of the host cell fuse. And then, so here's attachment, and then there's penetration with fusing. And then there's uncoating that occurs. And then once uncoating occurs, then the reverse transcriptase that rides along with the virus can then reverse transcribe the single-stranded RNA into double-stranded RNA. So this is really complicated. This is how it does it, and this is what I tell, when I teach. I don't use this particular slide, but when I teach graduate students, they have to know the viral replication of a retrovirus. But you just need to know that, or if you were in a virology class, a senior level virology class, you might have to know this. But you do have to know that reverse transcriptase makes double-stranded DNA from single-stranded RNA. Okay, so I would encourage you to go to the animations once again and look at HIV replication. So when you're done with that, if you want to listen to me some more, here's the HIV glycoprotein spikes binding to CD4 and CCR5, fusion of the virus with the viral membrane with the cell membrane. We have uncoating here, so now we have free RNA, reverse transcriptase that rides along with the virus, then can make double-stranded DNA from the RNA template. integrates it randomly into the host chromosome. It's probably not random, but we can have that discussion offline if you'd like. And then this is like a lysogenic bacteriophage. Now it's integrated into the host chromosome, and as long as the cell divides, it's going to copy its DNA. It's also going to copy the HIV double-stranded DNA in this case, the HIV proviral DNA. So once again, we have proviral DNA for HIV. We have proviral DNA or prophage for a bacteriophage. So those are two things that may help you remember, but you know, lysogeny, so there is kind of a lysogenic cycle of HIV. And then when the cell gets activated, and sometimes the LTRs are activated, they drive transcription, so we make viral RNAs, and then those viral RNAs come out of the cell, just like a regular polyadenylated 5' capped RNA. They pick up with a ribosome, they make viral proteins. Those viral proteins self-assemble, they get together with a viral RNA. The viral RNA goes inside of the virus and is released in, well, I guess so the viral envelope proteins are going through the endoplasmic reticulum into the Golgi, getting glycosylated, moving along here, where it picks up matrix protein, connects with the two copies of the single-stranded RNA, and then gets released from, or buds from the CD4 T helper cell. Now, when we talk about antivirals, we'll talk about some antivirals that work kind of right at this stage. So as the virus is released, it starts to mature, and protease inhibitors prevent that maturation. Okay, so this is text saying what I just said. One of the things that I didn't mention is that when the virions are released without cell lysis, so the cell can produce lots of virions, but sometimes what happens is as this GP120 is forming on the outside of the cell, it can connect with a CD4, a non-infected CD4 cell that might be right next to it, and form what's called syncytia. Okay, so these syncytia are multinucleated giant cells. Okay, I'm not really going to ask you about that, but just if you're ever curious and you take a virology class, a syncytium is a multinucleated giant cell. Okay, so now let's move to finish up with herpes viruses. Herpes viruses are very complicated. We don't understand a lot about herpes viruses, and you'll see how complicated they are in a second. They're big. They're 200 nanometers. They're twice the size of HIV. The genome size is, I think, about tenfold what HIV is. Yeah, HIV is about 10 kb, and herpes viruses are between 100 and 200 kb. 70 to 100 genes. I mean, think about HCV that only had like 7 to 10 genes. This has 70 to 100 genes. So there's a lot about herpes viruses that we don't understand. They do have an envelope, and the envelope is acquired by budding through the nuclear membrane. So in the picture I'm going to show you, it looks like it's coming out through the cell membrane, but the membrane actually comes through the nuclear membrane. So herpes viruses get a membrane from the nuclear membrane because they are a double-stranded DNA virus. So that's what it is. Herpes simplex is a double-stranded DNA virus. Two main types, HSV1 mainly causes oral lesions, HSV2 mainly causes genital lesions. And I know what you're thinking. We used to tell students that HSV1 was above the belt and HSV2 was below the belt. But we don't say that anymore. You can have HSV2 above the belt, and you can have HSV1 below the belt, and you can figure out how that happens since you're in college. So the first type of infection we have is an epithelial infection. So epithelial cells among the skin keratinocytes. Okay, So we have a latent infection that occurs in neurons. So generally this first infection is a lytic infection, and then once the virus, so once you have herpes, you always have herpes. It actually replicates episomally in cells. So herpes is forever. Chickenpox, for example, is a herpes virus, which is why you can get shingles, which is chickenpox the second time. So that's a herpes virus. Doesn't have to be HSV1 and HSV2. Okay, so no cure, no prevention for future outbreaks. So the structure here, there's a lot of complicated viral receptors here. We think that something called GB and GN are viral receptors for host cell receptors. This virus has a tegument, so a lot of proteins here that surround a capsid that encapsulates double-stranded DNA. Okay. Different cellular receptors, heparin sulfate might be one. Nectins, which are adhesion molecules, might be another. Once again, you can watch herpes virus replication on the animations page. But just quickly, we have a binding of the virus to a host cell. So let's say this is an epithelial cell on maybe your lip or skin, something like that. Then what happens is the membranes fuse. So we have the herpes virus membrane fuse with the cell membrane. And it kind of, when this happens, there's some microtubules that form here. And the virus capsid kind of rides down a microtubule elevator to the nucleus. And the reason it does that is because it's a DNA viruses. And DNA viruses replicate in the nucleus. So uncoating occurs here through a nuclear pore in the nucleus, and the DNA circularizes. Okay, once the DNA circularizes, then one of two things can happen. We can first have an infectious cycle, so we get immediate early, which means they found early genes first, and then they found some genes that happen even before the early genes, and they call them immediate early genes. So immediate early genes then are produced and from the circularized DNA. So this just uses the same mechanism. So a DNA-dependent RNA polymerase then would make this RNA. The RNA goes out into the cytoplasm, makes these proteins. These proteins are early proteins. A lot of them are regulatory. Okay, some of them help make a viral DNA-dependent DNA polymerase. So even though it's a DNA virus, it makes its own DNA polymerase. And actually, that's one of the vulnerabilities of HSV, is we can inhibit some features of that DNA polymerase, the herpes DNA polymerase. So the circular DNA replicates itself by rolling circle replication. You already know rolling circle replication. So we get concatenameric DNA here. Some late mRNAs are made from this concatenameric DNA. Late mRNAs also produce, as you remember, produce structural proteins. So structural proteins are produced. They actually go back into the nucleus, okay, and they form the capsid, and then they associate around the capsid with forming tegument. And then we have all these other viral surface. The surface of the virus is coated with all these other viral proteins, and this double-stranded DNA herpes virus buds from the nuclear pore, but it actually goes through the endoplasmic reticulum to do that, to help glycosylate all these proteins that are on the surface of the virus. Okay, and then it translocates through the Golgi, it gets glycosylated, as I mentioned, and then exits the cell. And there's lots of other things that are happening that that are in the animation. So this talks about what I just mentioned. Even though after 20 minutes this light shuts off, I'm going to continue going, and we'll be done with the lecture here in a second. So this deals with persistence in herpes virus infection. So herpes infects humans indefinitely, which means once you're infected with a herpes virus, you're always infected. One of the things I did not mention previously is this latent transcript. So we went through the lytic infection. Now, a latent transcript means we get a latent RNA, and that actually stops lytic replication. So we have a latency-associated transcript, and it makes latency-associated proteins, and the latent, or the LAT, or latent-associated proteins, and this is important, latent-associated proteins prevent the lytic cycle. Okay, so they allow the virus to be quiescent, and they allow it to be latent. Okay, so here we are right here. Herpes viruses can infect humans, followed by the initiation of latency by LAT, latency-associated transcript proteins, which suppress lytic viral replication. Okay, and that's generally what happens in neurons. Okay, so here we are here. We replicated in an epithelial cell, release the virus goes and infects a neuron, and then here is where we have a latent infection. Okay, so, but we could still have herpes virus outbreaks, or we can have varicella zoster, because old people like me, oh, I'm not that old yet, but I could get zoster. Actually, I have had zoster, which is chickenpox the second time, but I had it about 20 years ago. So stress brings that on. Sunlight brings this on as well, or a suppression of the immune system in any way. So then the latency-associated transcript is degraded and is not expressed, and we get a lytic infection that proceeds from the neurons back to an epithelial cell. Okay, as I mentioned, there are lots of different herpes viruses, a very big family. Cytomegalovirus is very common. All of you, probably except for one or two, are infected with cytomegalovirus that actually lives in cells in your bone marrow. If you become immunosuppressed, you'll have a problem with this virus. It's so ubiquitous. We already know varicella zoster causes shingles, okay? So if you heard about shingles, there's a vaccine for shingles. HSV1 and 2 we just talked about. Epstein-Barr virus, remember this causes infectious mononucleosis. So maybe some of you have already had this. Infects B cells. And HHV 6, 7, and 8 are problems in HIV patients and in some Mediterranean populations. Okay, so here's a clicker question. True or false, for influenza virus assembly and release, all viruses released have 8 segments of the influenza genome. Think about that for a second, pause the video, and that is false, because the virus, the influenza virus, doesn't care if it picks up 8 segments. It just hopes that it has 8 segments, and it hopes that it has the correct 8 segments so that it can be a productive infection.